All right, last time we talked about the cell cycle, all the parts of the cell cycle. You need to know those. You need to know G1, what happens in G1, S phase, what happens in the S phase, and G2, and what all three of them are called, interphase, and so forth. You need to know mitosis is nuclear division, and cytokinesis is cytoplasm division. And the phases of mitosis, the prophase, the prometaphase, the metaphase, the anaphase, and the telophase, and what happens in each one of them. I hope you went home and got that done, because before you know it, your next exam will be coming up on you real soon. So you need time with all this information. The cellular respiration, photosynthesis, those should already be done. But then you also need time with mitosis and today, meiosis. So mitosis and meiosis are both cell division cycles. Before I get into talking about the difference between the two, the last part of your chapter 12 talks about cancer. Real brief mention of cancer. And we talked about that. We said that checkpoint guards are checking to see if the cell is ready for division. And what did we say if in the scenario where the cell is not ready for synthesis of the DNA? Where does it go? G0. Good. What happens to the cell if it stays in G0 for too long when it's not supposed to stay there for too long? Apoptosis. Look at that. You've been studying. This is what I've been wanting from you the whole semester, is firing back at me. I fire at you with a question, you fire back at me with an answer. That's how you do it. Okay. Now, cancer cells disobey all those things. That's why they're cancer. Cancer cells don't know what apoptosis is all about. I mean, they're ordered to apoptose, but they basically respond by saying, no, I won't do that. Now you have a problem. Now you have cancer. Cancer cells behave really weird. And again, that's why they're cancer cells. They disobey certain rules. There's a rule that says this. If we are two cells growing next to each other and we run into each other, we're going to communicate together and tell each other that the reason we're running into each other is because we're running out of space. I mean, think about us here in this room. There's space between you and I. So the chances are we're not going to run into each other. But if you bring 3,000 people into this room, then we'll be hitting into each other, you see? But cells are very interesting. They say if we're running into each other, normal cells, we will stop dividing because we're running out of space. And in your chapter PowerPoint for chapter 12, they call that contact inhibition. When the two cells contact each other, mean run into each other, they're going to communicate to each other that we ran out of space, stop dividing. That's pretty clever. Guess what cancer cells do? They say, run out of space? Who cares? Just divide. If we have to even divide on top of each other, we'll do that, and we become a tumor. So cancer cells do not or lose contact inhibition. That's what happens. They lose contact inhibition. They get the message, but they ignore it. We're running out of space. Who cares? Let's just grow on top of each other and become a tumor. Now, there are other cells in our body there that they require to be attached to something for them to start dividing. So it's called anchorage dependence. Anchorage. It has to be anchored to something for them to start dividing. Again, cancer cells, anchor? Well, I don't need to anchor to divide. I'll just divide. So they can divide without that anchorage requirement. So look, cancer cells are cancer cells because they break or disobey the rules. And there are reasons behind that. They're genetically mutated, and we'll talk about that in another chapter. So they have genetic defects that prevents them from doing apoptosis, programmed cell death. They have defects that that prevents them from doing contact inhibition and from anchorage dependence. So they become a tumor. Now there are two kinds of tumors. There's the benign kind and there's the malignant kind. If you have to have a tumor, ask for the benign one, because that's not cancer. See, not all tumors are cancerous. There's the benign one, the non-cancerous one. And then there's the malignant one, the cancer one. The malignant one can undergo something called metastasis or metastasize. Let me see if I can spell that. Metastasis or metastasize. That's a fancy word for it can move around. It can go from one location to the other. Benign tumors, the less harmful ones, the non-cancerous ones, do not metastasize. The cancer ones, the malignant ones, they're malignant because they metastasize, because they move around. So a person may have breast cancer today, 3 months from now, they may have lung cancer or liver cancer also, if it's not taken care of before the cells metastasize. Because once they start moving, it becomes a fight between your doctor and the cancer. And guess who normally wins? Unfortunately today, the cancer wins eventually. Depends on, of course, the type and how aggressive and when did you first notice it and so forth. So of course there's a lot of research going on in cancer today to trying to alleviate this problem. It's a major killer around the world, not just in the United States. But what is cancer? What is a tumor? It's simple. It's uncontrolled cell division. If someone asks you, can you tell me what cancer is? It is uncontrolled cell division, meaning the cell cycle broke down somehow. See, it is so amazing. The cell cycle is so amazing. Of the intricate control of the cell cycle is phenomenal. You think cellular respiration was tough. When you go and take an advanced class in biology, if you do, and they start teaching you about all the intricate detail of how the cell cycle is regulated, you will know why it can easily break. Nowadays, our cars, a long time ago, our cars, they were easy to fix. They didn't have too many gadgets in them. And some people actually learned how to fix their own cars so they can save money. Can't do that today. Every little thing in your car nowadays is sophisticated to the point where if one thing breaks, you look at it and say, I can't touch this. I don't even know what that is. Let me take it to someone who actually knows. Because they're so sophisticated nowadays. Cells, or the cell cycle, is similar to that. There's so many moving parts. The regulation is phenomenal. So it makes sense that one little thing breaking in it messes up the whole thing. And that's why cancer is so difficult to figure out, because not all cancers are caused by the same defect. And some are more aggressive than others. Okay, so this is my basic quick discussion on finalizing chapter 12. They do talk about cancer at the end of the chapter, so you need to know what metastasis is. This is when a malignant cancer cell moves from one location to the other. You need to know the difference between a malignant tumor and a benign tumor. The malignant tumor is the cancerous one. The benign one is the non-cancerous one. You need to know what cancer is all about. Cancer is uncontrolled what? Cell division. You also need to know that cancer cells are basically defective cells. They're actually called transformed. Here's another word for cancer, by the way. A transformed cell is a cancer cell. Transformed means changed. a cell that changed to become a cancer. That's another word for cancer or tumor. Transformed cells. So what transformed about them? There's some kind of genetic defect that basically prevented them from regulating their cell cycle properly. There's so many different reasons behind that. But once they become transformed, they can just become benign or malignant. Okay? So what this allows me to do now is to start talking about the differences and maybe the similarities between mitosis and meiosis. The next chapter is meiosis. In mitosis, when we talk about mitosis, we're talking about a parental cell. We're only going to talk about humans. So our parental cells will have 46 chromosomes. So we're talking about mitosis first, and we're going to do a quick comparison between mitosis and another kind of cell division called meiosis. Both of them will start with a cell that has 46 chromosomes. Again, we're talking about humans. We're only going to talk about humans the rest of the semester. 46 chromosomes. The 46 chromosome number is called our diploid number, which is also designated with 2 little n. And our diploid number is 46 chromosomes. Or you can say 23 chromosome pairs. Pairs, that means 2 of each. You do have 2 of each chromosome. You have one from mom and one from dad. You have one complete set from mom and one complete set from dad, and you get that during fertilization. So mom gave you has the egg component with 23 chromosomes, and dad supplied the 23 other chromosomes on the sperm. And at the point of, that's called fertilization, when the sperm fertilizes the egg. And what ends up from there is the fertilized egg, 46 chromosomes, and that's called the zygote. So you are half mom, half dad. You have a complete set of chromosomes from mom and another complete set of chromosomes from dad. And they were put together at the point of fertilization. And this is basically you at the point of conception, a zygote. Yes?
I have a question. I don't know if it's related to this, but let's just say that someone want to have a child and you cannot have one, and you find somebody to use your sperm and then somebody else. So that's kind of like 3 people. So how many?
No, you're still a product of one egg and one sperm.
But somebody else.
But it's somebody else, yeah. It's a donor.
Somebody else's DNA.
It's like an unknown dad.
Then I'm saying somebody else carrying it.
No, like she's
A third person carries it.
Yes.
That is a surrogate mother. That you still only have 2 copies because she's just the incubator. The carrier is just the incubator. She's not going to be providing DNA. Yeah, the female surrogate mother, that you would have to pseudo impregnate her and all that. It's a complicated process. But if someone is carrying the zygote for someone else, that person is not contributing to the DNA. Just it becomes an incubator. Okay, so you are now going to be produced from this through mitosis. So mitosis made you who you are today. It developed you as a baby. You grew after that, and you became a teenager and all that. But eventually, you will start producing sex cells. For females, it's going to be eggs. For males, it's going to be sperm. So at the point of puberty, you will be producing what's called sex cells, which is also called gametes. That is by meiosis. So meiosis will produce the gametes. But they are produced from this cell. So a stem cell is going to be used to make the gametes. And again, that would be either sperm for men, eggs for women. Now, all your other cells multiply by mitosis. Now look at this, short list, long list. Remember the what list?
Short list.
Everything else is on the long list. Here's your short list. Gametes are sperm and eggs produced by meiosis. That's it. All your other cells are called somatic cells. For example, liver cells, somatic. Skin cells, somatic. Heart cells, somatic. Anything that is not egg or sperm is a somatic cell. And that is by mitosis. So let's put it together. Somatic cells are divided by mitosis. Gametes are produced by meiosis. Somatic cells are diploid. The gametes that we're going to produce at the end will be haploid. We'll only have N, which is 23 chromosomes. Look at the difference between the two. This is 23 chromosome pairs. That means 23 times 2, which is 46. For the gametes, it's just 23 chromosomes. So, what do you think? This egg right here, is that haploid or diploid?
Haploid.
Haploid. This sperm?
Haploid.
Haploid. The zygote?
Diploid.
Diploid. Good. How were these produced?
Dividing.
Meiosis. How will this divide until it makes you who you are today?
Mitosis.
Mitosis. So the zygote is diploid because all you have to do is look at the chromosome number. It's 46. This is the egg, and that's haploid. This is the sperm, and that's haploid. Look at that. Meiosis is designed to bring the chromosome number down to 23. The goal of meiosis is to take this 46, bring it down to 23. And that would be the egg, for example, in females. In men, same thing is happening. Bring the 46 down to 23, but then there'll be sperm. And then when do I get the 46 back? At fertilization. Yeah.
Yeah, so the gamete cells, they don't divide. They just discard.
No, once they're made, they're designed for baby to make a new life. So if they're not used for that purpose, they get recycled. For example, the egg that's the menstruation and all that. So it gets removed, discarded. But once it's fertilized, once the egg is fertilized, it's now gonna have its own program of developing a baby. Not all eggs get fertilized, of course, but the ones that do develop a baby. That's what they're designed for. So the trick is meiosis drops the chromosome number down to half, and then the diploid number comes back. At the point of fertilization, you're now diploid at 46. So if we look at the grand view of mitosis here, we realize something here. Mitosis produces two daughter cells that are genetically exactly the same to each other and to the parental cell. We talked about that. Mitosis produces two daughter cells that are exactly alike to each other and to the parental cell. Can't say that about meiosis. Meiosis is different. Meiosis actually has two phases, two meiotic events, meiosis 1, meiosis 2. The first meiosis 1, look at this. The first meiosis 1 will produce two cells with 46 chromosomes in them, but then each one of them will multiply again. But then there'll be half of 46 in each one. 23, 23, 23, 23. You'll end up with 4 haploids at the end of meiosis. So you can tell there's a difference here. There's actually 2 events, meiosis 1 and meiosis 2, compared to 1 event here. And that would be mitosis. But the name of the events are pretty much the same in your book. They have it as Prophase 1, Metaphase 1. Notice there's no pro-metaphase between them. Anaphase 1, Telophase 1. That's in meiosis 1. In meiosis 2, can you guess? Prophase 2. Metaphase 2, anaphase 2, telophase 2. And each one of them is followed by a cytokinesis event. That's why they split into 2. Okay, so we talked about these 2 daughter cells being the same as this one and the same as each other. Can you tell me something? Are these 4 cells, haploid cells, the same as this one?
No.
How do you know that?
They have less chromosomes.
There you go. It is so obvious that they're not the same. These 4 haploid cells, the cells that you're going to end up with, no way that they're the same as this one. No way. All you have to do is look at 46 versus 23. Here's the interesting thing that you can't see, and this is what we're about to talk about. They're also different from each other. Each one of these will also be different from each other. Can't see that yet, but you will. That's the goal of meiosis. The goal of meiosis is, number 1, drop the chromosome number by half. Number 2, genetic variation. That's the goal of meiosis. Look around you. You are all products of meiosis. Are there 2 people in the room that look the same? No. So meiosis succeeded. Meiosis produced genetic variation. And all I have to do is look in this room here. No 2 people are the same. Of course, the only ones that would be like that would be identical twins. And we'll talk about how identical twins are produced versus fraternal twins. All 4 haploid cells are different from each other. So I am one of 4 children. This would be my brother, my sister, my other brother, and that's me. I'm the youngest one. So we're all different from each other. We share some similarities, of course, because we come from the same mom and dad. But we're also different from each other. That's the goal of meiosis. The goal of meiosis is genetic variation, yeah?
After the first phase of meiosis, is that called are they called daughter cells? The 2, yeah.
These guys here.
Okay.
And here's the interesting thing. These are also different from each other.
So they're not.
I wouldn't call them even daughter cells,
Okay.
yeah. These are daughter cells. They're really identical to each other and to the parent. For the meiosis, genetic variation starts very early on in prophase 1 here. We'll talk about it. So they're right away different from each other. So I would call these haploids. They're also technically these are also called haploids, but these are for sure haploids. We'll talk about why that's the case. Okay. We said, listen to this one please. We said that during the cell cycle, the DNA duplicates. Do you remember that? In which phase?
Synthesis.
In the synthesis, S phase. So in the S phase, this one went to 92, remember that?
Yeah.
And then it went through mitosis and then divided into 46 and 46. So there is an S phase, of course, right before you get into nuclear division in mitosis, and we talked about that. Well, the same thing is here. There's an S phase right before this, where the 46 will become 92. And then they're gonna go through this, and then they're gonna split 46 and 46. But catch this one. There's no S phase in meiosis 2. If there was S phase in meiosis 2, how many chromosomes would you have here?
46.
Yeah, then you would defeat the purpose. The purpose is to half the number of chromosomes. So look, 46 double to 92. So each of these can be 46. But then there's no S phase. There's no S phase here. It's just nuclear division again. So then you end up with 23 and 23. That's how. Follow the numbers. Okay. Now, what we're about to do is show you the events of meiosis. What's happening here? What's happening here? Here's the interesting thing. In meiosis, the same events that happen in mitosis happen in meiosis. So for example, in prophase, the nuclear division begins to disintegrate. Yeah, that happens twice in meiosis. In prophase 1 and prophase 2. In prophase, the DNA becomes more visible. Yeah, that happens twice in prophase 1 and prophase 2 in meiosis. The chromosomes line up at the metaphase plate. Yeah, that happens twice in metaphase 1 and metaphase 2. What I'm trying to say is, I'm not talking about those things anymore. I already did on Monday. The events that happen in mitosis are a lot similar to the events that happen in meiosis. It just happens twice. So you need to go back and review what happens here and compare it to what happens here. What I'm about to talk about, however, are those things that are different from mitosis. The differences between meiosis and mitosis. For example, in mitosis, you had one chromosome, one from mom, and one chromosome, one from dad. They duplicated, and then at the end, each one of these got one of each. So they are identical to each other and to the parental. That's not going to happen here. It gets a lot more complicated. What's going to happen is this. I use my fingers to illustrate this for you. And a lot of students like this here. Now you'll have it on video for you to follow through. I'm going to follow this one first, and it'll be so easy. You'll be like, I understand this one. But then we'll do this one and show you how complicated, how the difference is. Dad's chromosome 1, mom's chromosome 1. Here they are. They duplicate in which phase?
S phase.
In the S phase. It's okay. They find each other. In which phase? They find each other and attach at the centromere. In which phase?
M phase.
The first phase of mitosis. In prophase. Let's do it again. Mom's chromosome 1, dad's chromosome 1. They duplicate. In which phase? S phase. They find each other. In which phase? Prophase. They line up. In which phase? Metaphase. They split. In which phase? Anaphase. We're done. That's mitosis. Let's do it one more time because I need you to understand what I'm doing here with mitosis. It's going to get more complicated with this one, as you can tell. Mom's chromosome 1, dad's chromosome 1. Of course, you have 2, 3, 4, 5, 6. But I only have so many fingers, right? They duplicate in which phase? S phase. So you imagine the cell going to the S phase and the DNA being copied. Good. Now these are chromatids of each other. Am I right? More specifically, sister chromatids, sister chromatids. Use the words. They find each other and attach at the what? Centromere. During which phase? Prophase. Good. They line up. Which phase? Separate. Now what are they called now? Okay? No, they're sister chromatids right here, but once they separate in anaphase, they are daughter? No. We're talking about the chromosomes. Daughter, nah. Daughter chromosomes. Let's do it one more time. Chromosome 1 from mom, chromosome 1 from dad. They duplicate in which phase? S phase. They come together, attach at the centromere. Which phase? Prophase. They line up. Metaphase. Right now they're called chromosomes, but they're made up of 2 sister chromatids. But once they do this, in which phase? Anaphase. Now each one of them is called a daughter chromosome, because we're following the chromosomes with the fingers there. Let me show you how this one is different. Everything that I'm about to do right now, here's the cool thing, happens in Prophase 1. So when in doubt on the exam, say what? Prophase 1. Now hopefully you don't do it with doubt throughout the whole exam. But let's say you're like puzzled, and let me tell you something. If it has to do with genetic variation, it's Prophase 1. Because what we're about to do is shuffle the genes. This is no different than if you have played, what's that called? Blackjack in the casino, where they come with cards, right? The blackjack dealer will come with a deck of 7 or 8 decks of cards, and they shuffle them. It's no different than that. But you're not shuffling cards, you're shuffling your genes. That's why these are going to be different from each other. No 2 shuffles will be the same. Or what are the chances that the shuffle is the same? Very, very little, especially when they're dealing with 7 or 8 decks of cards. Good luck trying to shuffle 8 decks of cards and another person randomly shuffles another 8 decks of cards. Go ahead, compare the shuffles. What's the chance of both of them at randomly being the same? Very, 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 very little. Which is the same idea here. You're shuffling the genes. That's why these are going to be different from each other at the end. I'm going to show you how to set up the shuffle and then to actually do the shuffle of the genes. So the blackjack dealer goes into the back of the room, gets 8 decks of cards, opens them up at the table so make sure everybody sees them, and then he sets them up and he starts shuffling them. So he's getting ready, and then he holds the 2 decks and he goes like this, and then he shuffles. So all the events that he did before he held them like this is getting it ready for that shuffle. But once he lets them go and they go like that, now they're shuffling. No different than here. I have to prepare for the shuffle and then go ahead and shuffle. So here's what we're going to do. We're going to follow again chromosome 1 from dad and chromosome 1 from mom. But now it's meiosis. Chromosome 1 from dad, chromosome 1 from mom. They duplicate in which phase?
S phase.
In S phase. So that is not different. They find each other, attach at the centromere. In which phase? There is no such thing as prophase in meiosis. What is it?
Prophase 1.
It's called Prophase 1. Watch out for that on the exam. When you say prophase, I'm going to assume you're talking about mitosis. Why? Because there's no prophase in meiosis. It's called either Prophase 1 or Prophase 2. And on a multiple choice exam, they're going to throw you a loop or a trick like that. Prophase 1, prophase 2. So get used to it. When you're talking about meiosis, you have to add the 1 or the 2 after the name, so you get used to it. They do find each other and attach at the centromere in prophase 1. Now watch this. At this stage, look up here please. These are sister chromatids of each other. Do you agree? These are sister chromatids of each other. But these are non-sisters. And these are non-sisters. Let's do it again. Dad's chromosome 1, mom's chromosome 1. When they duplicate in S phase, what did they create? Sister chromatids. But these are that's dad's 1, and that's mom's 1. That's why they're sisters of each other. But these are not sisters of each other, because that's dad's 1, and that's mom's 1. So the 2 dad's chromosome 1s are sister chromatids. The 2 mom's chromosome 1s are sister chromatids. But the one from dad and the one from mom are non-sister chromatids. Then when they do this, that's now called the homologous chromosome. Homologous to this one, meaning this is chromosome 1 and that's chromosome 1. So in prophase 1, you have to form the homologous chromosomes. You're going to have 2 homologous chromosome 1s. One from dad and one from mom. And each homologous chromosome is made up of sister chromatids. Are we catching the words? You're going to have 2 homologous chromosome 2s. One from dad and one from mom. And each homologous chromosome is going to have 2 sister chromatids. And all of that is happening so far in prophase 1. Watch this. The 2 homologous chromosomes will synapse. Watch please, look up here. They will synapse. They will do this. It's like Velcro. Watch. Velcro. There we go. So this is synapsing. They come together. Synapse. So they're gonna do synapses. The coming together. Who's synapsing the homologous chromosomes? So the 2 homologous chromosomes for chromosome number 1 will synapse together. All of that again is in prophase 1. I have not left prophase 1 yet. So the sister chromatids come together, the homologous chromosomes come together and synapse. They attach to each other. Now, I don't make these words. I'm just a messenger. There's a name for the Velcro. It's called the synaptonemal complex.
Oh.
I know.
I guess it's close. So.
That's the Velcro. That's the sticky material. How's that? So when the 2 homologous chromosomes synapse and attach at the synaptonemal complex, look at what they form. They form 4 of them, right? That's called a tetrad. Tetra means 4. So they form a tetrad. Again, all of that is like the blackjack dealer getting ready to do the shuffle of the cards. You have not shuffled anything yet. They're just getting them ready. They're taking the cards out of the boxes and putting them on the table and getting it all ready. They haven't done the shuffle yet. We haven't done the shuffling yet. We're just getting them ready. Let's do another review. You shout it out now. Mom's chromosome 1, dad's chromosome 1. They duplicate in which phase?
S phase.
S phase. They come together. Now we're talking about meiosis. They come together. They form what of each other? These 2 are what of each other? Homologous chromosomes. But watch. Look. What are these of each other?
2 sister.
Good. What are these of each other? What are these? What are these? Ah, nice. So if you pay attention real close, look, it's the non-sisters that are attaching together. Look at it. There are 2 non-sisters on the inside and 2 non-sisters on the outside. Do you see that? That's important. What is this coming together? What is that? Synapses. What's the attachment point? Synapto-nemal complex. And what is this structure now? The 4. What is it? Tetrad. Are we okay so far?
So, yeah, okay, so the tetrad is just the 4 chromosomes?
The 4, yeah.
What's that?
Tetra, yeah. Tetrad 4, yeah. Now watch this. The 2 non-sister chromatids on the inside, if you're watching up here, will form this structure. I did not do this. I said the ones on the inside will do this. The ones on the outside will do nothing.
Still, prophase 1. Correct.
Oh yeah, so far all of that is in prophase 1. This has a name. It's called chiasmata.
It's like a gang sign. Sorry.
It's like building your vocabulary, right?
Yes.
All of that is prophase 1. So if I have to say it in words, if my professor forces me to get up in front of everybody to describe this event, here's what I would say. Follow. Dad's chromosome 1, mom's chromosome 1, they duplicate in the S phase. They come together, attach together through synapses in prophase 1. The homologous chromosomes attach to each other by synapses in prophase 1. The attachment point is called the synapto-nemal complex, forming a tetrad. The non-sister chromatids on the inside will form a chiasmata. At this point, that's when the blackjack dealer is holding the cards like this, and he's about to let them go. He's about to shuffle the deck.
Isn't that called crossing over?
Yeah.
That's what it's called?
This is the structure is formed. The structure formed is called chiasmata, but the process is called crossing over. The structure is chiasmata, but the event is called crossing over. And it has nothing to do with anybody dying here, right? This is crossing over of genes now, right? All right, now, the crossing over part would involve a lot of pain in my fingers there, so I'm not going to do that. But imagine me doing this. I take this finger off, half of it, and this, that would be painful for me. I love my fingers, so I'm not going to do that. And this finger off and exchange them. That's the shuffling part. Now we shuffle the genes. And that's the crossing over part. So after the chiasmata is formed, this part will go to this part, and this part, they'll do a shuffle, a gene shuffle. Now I'm only showing you one crossing over event, but along the length of the chromosome, like so for example, chromosome 1 is your longest chromosome. You can have crossover events all the way through it. Now, after this prophase 1, what you have is now, imagine this finger here is red, and this one is red, and this is blue and blue. After you've shuffled them, this is still red, because this one did not do the shuffle. This one did, right? This is still red, but now what about this one? Is it still red? Blue and red. What about this one? Blue and red. What about this one? Blue, right? So the inside ones are the ones that do the gene shuffling. The outside ones don't. Now the rest of meiosis is to separate these. Watch. After I've done the shuffle here, the rest of meiosis is to do this. Go through meiosis 1 to separate these two. Go through meiosis 2 to separate these. So you do the shuffle in prophase 1, and the rest of the event is dealing with separating them.
So it's the same as mitosis other than prophase.
The rest of it is the same. Now the difference here is this. In metaphase here, mitosis, these sister chromatids that are now called chromosomes are lining up. Watch that. In metaphase 1 here, it's the tetrads that are lining up. See, after I do the shuffle here, what's going to line up in metaphase 1? It's the tetrads. The shuffled tetrad for chromosome 1. The shuffled tetrad for chromosome 2, 3, 4, 5, 6, 7. And they don't have to line up in order there. They don't know that they're called number 1 and number 2. They can line up at random there. So in metaphase 1, the tetrads are lining up. Here, it's the chromosome that's lining up, but it's made up of 2 sister chromatids. You gotta catch the words here. In mitosis, the chromosomes are lined up. Each one of them is made up of 2 sister chromatids, and when they separate, what did we call them? Daughter chromosome. That one is easy. But for meiosis, in metaphase 1, it's the tetrads that are lining up after they did the shuffling. Tetrad for chromosome 1, 2, 3, 4, 5. Now watch this. In anaphase 1, who's separating? Who's separating?
The sisters?
Tetrad.
The homologous chromosomes. I mean, it's weird that we call them homologous. They're only homologous because they're both chromosome 1s, but they're no longer the same. You know that, right? Because they've shuffled. So look who's separating. In anaphase 1, it's the homologous chromosomes that are separating. Look at metaphase 2. In metaphase 2, it's the homologous chromosomes that are lining up. Good. Anaphase 2, who's separating? Yeah, the sister chromatids. Want me to do that again?
Yes.
I think so, yeah. So, in prophase 1, the tetrads form, the chiasmata forms, and the crossover happens. Now you have shuffled genes. And that next is my metaphase 1, the tetrads line up. In anaphase 1, the homologous chromosomes are separating. In metaphase 2, the homologous chromosomes line up. In anaphase 2, the sister chromatids are separating. So if we follow the color scheme, let's say, for example, you had dad's chromosome 1 is blue, mom's chromosome 1 is green. By the end of S phase, you will have 2 green ones and 2 blue ones. And then you're going to get into prophase 1. Look what happens in prophase 1. A whole bunch of things happen. All 4 of these come together. That's in prophase 1. And then a chiasmata forms. Watch this. A chiasmata forms. And then the crossover happens. I have to think when I do this here so I don't make a mistake. Green, partly blue, blue, partly green, and then another green one and another blue one. See that? That's the crossover. That's equivalent to me taking a piece of my finger and exchanging it there. In metaphase 1, that's what's lining up. The crossed over tetrads. In anaphase 1, those guys are going to be separating, so you're going to end up with this. Like that. So that's like equivalent to me doing this. One of them has a full green one, mostly green, partly blue. Full blue one, mostly blue, partly green. And then these are the guys that are going to be lining up in metaphase 2. These homologous chromosomes inside this cell is going to be lining up in metaphase 2. And then they're going to separate in anaphase 2. So look what's going to happen. You're going to have a full green one coming from this one, and a mostly green, partly blue. Full blue one coming from this one, mostly blue, partly green. Did I prove to you that the 4 haploid cells are different from each other? Actually, they are. At the chromosomal level, at the DNA level, this one is not the same as that one, is not the same as that one, is not the same as that one. Genetic variation. And that's what meiosis wants to achieve. Meiosis wants to produce haploids that are different from each other. What's the purpose or the goal or the benefit of that? Survival of the species. With genetic variation, we have a chance to survive the everyday assault of us going extinct. I mean, we're under a lot of pressure here, but we're still here because of genetic variation. No, so nature wants this. Okay. Now, there are a couple of events that happen that ensure genetic variation. Genetic variation is so important. Of course, meiosis, the products of meiosis obviously are different from each other, and that guarantees genetic variation. So when does the variation begin? Think about, this is a critical thinking question now. Based on what I said, when does the variation begin? At the point of what? No. Say? Say it. Yes, crossing over. You can say, when are the cards shuffled? When they're shuffling. During crossing over, that's when variation begins. So, specifically, Prophase 1. I told you everything or almost everything that has to do with shuffling the genes is in Prophase 1. The next event, Metaphase 1, factors into that too. I told you that there's no set way of these tetrads lining up at the metaphase plate. I mean, we name them chromosome 1, chromosome 2, chromosome 3. They don't know they're chromosome 1, 2, or 3. So when they go and line up to begin the race at metaphase 1, they don't know who's next to them and who's... They just go to line up. And then sometimes this guy lining up is facing this way, sometimes maybe facing that way. Know that if it's facing this way, then this one is going to this cell, and this one is going to this cell. Okay, what if it lines up this way? Then this one is going to this cell, and that one is going to that cell, you see? So at the end, you're gonna end up with a whole array of things. This one might have the full chromosome 1 from mom, but then the crossed over one, chromosome 2 from dad, and so forth. It's all random. So the lining up of these tetrads in metaphase 1 and how they separate in anaphase 1, they're lining up in metaphase 2 and anaphase 2 separating. All of that factors into additional genetic variation, because you end up saying, I cannot predict who's going to go where. And that factors into genetic variation. So the shuffling, the lining up of these homologous chromosomes and the tetrads, and then when they go to separate, you can't predict which one is going to go where. And the fact that we're talking about not just one crossover event, we're talking about hundreds of maybe thousands of crossover events within one chromosome. We're only looking at one crossover. Chromosome 1 would have crossovers all over the place. Oh, there's a lot of shuffling going on. Guarantee that this one will be different from all three of these. Here's another layer of genetic variation. The third layer of genetic variation happens at the point of fertilization. You cannot control which egg gets fertilized by which sperm. I know you might take that as an insult. Wait a minute, I'm married. I have a boyfriend, whatever. Regardless of whether you're monogamous or not there. You cannot control which egg is fertilized by which sperm. Even if you do it in vitro, it's still a random event. So we call it random fertilization. So the crossover event factors into genetic variation. The fact that these chromosomes independently assort from each other. Chromosome 1 here ended up here, independent of where the other ones ended up. It's called independent assortment of chromosomes. Major component of genetics. We'll talk about it in more detail. Just know what it is right now. We're talking about the events that ensure genetic variation. Crossovers in prophase 1. Oh yeah, that will increase genetic variation for sure. How they line up and how they move into the haploids independent of each other, independent assortment of chromosomes. That will ensure variation. And just to add another layer of genetic variation, random fertilization. You have no control over which egg gets fertilized by which sperm. Doesn't matter if you're married or not. So why is genetic variation important? Again, for our survival. So what do we do as humans? We try to clone people. Cloning is against nature. Nature wants genetic variation. Cloning goes against it. There is no benefit to cloning a human being other than the scientist saying, I did it, and an ego thing. Biologically speaking, there is no benefit to cloning Dr. Saad. It's actually really, really bad for you. Because in 2 or 3 weeks or whatever, you're gonna have 100 exam 3s, not just one. Think about that one. Now there's no benefit for sure, right? So if you have 1,000 copies of me, in the big grand scheme of things, that is not good. Genetically speaking, it is much better to have genetically variable, genetic variation, than to have a whole species of one set of genes. Because when you have a stress event, let's say you have an island full of Dr. Saad, and the environment changes on that island where my genes, they don't work well with the environment, then all the people on that island, they're all gone. Because we don't have the variation for natural selection to occur so the species survives. So some of them, we just clone more, yeah. So some will survive, but others won't.